everyone. Today I'm going to talk about Module 18, which is on vision. So when we're talking about vision, we have this incoming light. So the wavelength is the distance from the peak of one wave to the peak of the next wave, and this determines the color. So a high wavelength is associated with colors on like the blue spectrum, and when we're talking about wavelengths of sound, it is more high pitched. Whereas a low wavelength is associated with red colors and um, lower pitched sounds. And wavelength is related to frequency. So, frequency is the number of waves per second. And if we have shorter waves, that's going to be more waves per second. So, a low wavelength is associated with a high frequency, and vice versa. Next, we have amplitude, which is the height of a wave. So, the distance between the trough and the peak. And this determines the brightness of a color. So, the brightness of a color and then the volume of the sound. So, high amplitude is going to be bright colors and loud sounds, and low amplitude is going to be more dull colors and softer, quieter sounds. Next, let's talk about the pathway that light travels through our eye and how this ultimately translates into what we see. So, first we have the cornea, which is this protective outer layer that bends and kind of refracts the light back onto our retina. And this kind of helps us to focus. If it's the wrong shape, that could cause near or farsightedness, which they can sometimes fix it with LASIK surgery, but they don't like to do this until you're like 25 or so because when you're younger, your vision tends to change a lot. That's why kids and teens need to like go to the eye doctor more frequently because our eyes just change a lot more at this stage. And if the cornea is the wrong shape, it could also cause astigmatism, which is it doesn't refract properly, so it gives you like two focal points, and then it makes your vision more blurry and can also cause like lights, especially at night, to kind of like look weird and have like halos or like little flares coming out of them. Um, yeah, after the cornea, we have the pupil. And the pupil isn't actually a structure, it's just like a hole. And it adjusts, the hole adjusts like how big it is to let in the perfect amount of light so that the picture isn't washed out but it's bright enough to see. Next is the iris, and this is the colored part of the eye, and it's the muscle that actually controls how large the opening or the pupil is. So the iris and the pupil kind of work together to make sure that the right amount of light is getting in. And interesting, you can tell someone's identity just by their iris, kind of like a fingerprint. It's very specific. Um, and people's eyes dilate when it's dark in order to let in more light. Also, when they're about to answer no to a question or when they're feeling disgust. And sometimes at the eye doctor, they'll dilate your eyes with like these drops so that the pupil gets big enough so they can take pictures of your retina. And this makes sure your retina's okay and also can like sometimes detect other diseases like diabetes before other signs show up. Next, we have the lens. So this is a clear fluid structure about the size of an M&M, and it changes shape to allow you to focus. It's kind of similar to the cornea. So if you like put your finger in front of your eye and look at your finger and then look at like something behind you, like the wall behind you, then look at your finger and like switch your gaze back and forth, you'll kind of feel your eye moving, and that's the lens changing shape in order to focus correctly. And that is actually called accommodation. Um, so, yeah. And interesting that when the light gets like through the lens, it kind of goes upside down, so it makes the picture upside down. Um, yeah, but luckily the brain is fixing that later on in the process. Some problems with the lens: if it's the wrong shape, you could have nearfarsightedness, need glasses, also astigmatism, need toric lenses, and if it's cloudy, that's cataracts. And a quick description of acuity. Um, so I'm pretty sure you've heard 20/20 vision. That just means that you can see from 20 feet away what the average person can see from 20 feet away. Um, if you have worse than that, like if you have 20/40 vision, that means that um, you can see you can see from 20 feet away what the average person could see from 40 feet away. Or if you want to flip it, what the average person could see from 40 feet away, you won't be able to see it until it's 20 feet away. Um, so yeah, if your acuity's not good, you probably need glasses. Some people with glasses or just naturally have better acuity than average. Like they could have 20/15 vision. So what the average person can see from um, sorry, let me get this right. What the average person can see from 15 feet away, this person can see it from 20 feet away. So it's extra good. Birds have like 25 vision. So what the average human could see from 5 feet away, a bird could see it all the way from 20 feet away, which is great for their, they need like super good vision for like hunting and stuff. Yeah, so next is the retina. So up to this point, we've had like all these steps that are kind of like passing the light through. So the cornea refracts it, the pupil and iris make sure the correct amount of light is going through, the lens also refracts it and kind of like centers and focuses the image onto the retina. And the retina is this thin paper like structure at the very back of our eye that actually interprets the light that has been kind of put reflected in. So it actually processes the light that has come in. And because it's like the processing center and kind of connects to the brain, this is how that image that was flipped upside down by the lens gets put right side up, because our brain, it's not like a whole picture, it's more like these individual cells in the retina sending messages to the brain, and that occipital lobe and visual cortex interprets everything. So the retina has two types of cells. It has rods, which are on the periphery of our vision, so helps us see to the sides, and rods kind of detect like black, white, and gray, not really colors. They're better with night vision. Yeah, and the cones are the other type of cell, and they do better in well lit conditions. They are toward the center of our vision, and they kind of are more better for like fine detail and color. So, the center of the retina where all the cones are is called the fovea. So it's kind of interesting. You will see yourself that your central vision is more fine detailed and accurate than your peripheral vision. And also, if you look at colors from the side, it's like not as accurate. So that kind of shows you where your cones and rods are. The cones are in the center, seeing the fine details. Yeah. So up to this point, all of this has been going from the front of our eye to the back. Now, for some reason, it kind of flips. So from the retina, we're going to the bipolar cells and the ganglion cells, and this kind of like process from cones and rods to bipolar to ganglion cells, this is going back toward the front of our eye. Not quite sure why, um, but yeah. And so a difference between cones and rods here is cones are kind of like soloists. So you got like some direct pathway from like one cone to one bipolar cell to the next ganglion cell. Um, but rods are operate more like a chorus. So a bunch of rods all send to one bipolar cell. And this is kind of why the cones have more fine detail, because they're more directly sending these impulses to the brain. And this is also why we have a lot more rods than cones. So we have like 6 million cones and 120 million rods. So after we pass this information from this kind of like relay from cones and rods to bipolar cells to ganglion cells, it gets to the optic nerve. And the optic nerve then sends it to the brain. So something cool about the optic nerve is where it like connects at the back of our eye to the retina. There is like a blind spot there because there are no receptors where the connection is, which makes sense. So everybody has like a blind spot in both of their eyes just because there's no receptors there. But it doesn't usually matter that much because we're moving our eyes a lot and the other eye can like see where that C in that blind spot. So it kind of like fills in the picture. Yeah. So from the optic nerve, we go to the occipital lobe, visual cortex, and that is where all of the interpreting of our vision happens. So that is pretty much it for the structures of our eye and how they all work together to enable us to see. Couple more things with the processing of this visual information. We have feature detectors, which are nerves in the visual cortex that kind of interpret different shapes, angles, or movement. We also have an area in our right temporal lobe that recognizes faces, and when it's like stimulated in experiments, it kind of like causes people's faces to just appear to like morph into other faces, which sounds crazy. And then some people have facial blindness where they cannot tell who a person is based on their face. And it's cool because through brain scans, they can tell like what somebody's looking at just based on the activity in their brain. Also, our brains are really crazy. They process all these things through parallel processing. So they actually process form, depth, color, and movement all simultaneously but separately, and then put it together, which we wouldn't really know that because to us it seems like it's just all in one, but our brain is actually like putting together all the pieces like really quickly. Next we have some theories on color vision. So young Helmholtz trichromatic theory says that the three primary colors of red, green, and blue, we have these cones that are designated. So there are cones designated to red, cones designated to green, and cones designated to blue. And with combinations of these colors, that is how we are able to see the whole spectrum. So they say that color blindness is caused when one of these like types of cones or few types of these cones are not working. There is also the opponent process theory, which is by Herring, and this one says that there are three sets of opposing colors. So there's red opposes green, blue opposes yellow, and white opposes black. So this theory says that this kind of opposing color thing is what enables us to see color. So, like, certain cells or receptors are stimulated by red and inhibited by green, and certain cells are stimulated by green, inhibited by red. And this kind of explains this idea of an after image. So if you like stare at something red for like a while, and then you look at a white paper, you'll kind of see that there's like a little bit of, it'll look kind of greenish. So um, the idea is that you look at the red paper and you tire out your like red receptors, and then when you look at a white paper which has all of the colors in it, because the red receptors are kind of tired, you see green. So yeah, both of these theories, I guess, have something to them and can simultaneously explain how we see color.